name is Sward, and I'm the field application scientist for Geneyx. And today we're going to be talking about how we can automate clinical workflows using our platform. I always like to give just a general introduction into the webinar. So if you have any questions throughout the demonstration, please feel free to type them into the chat panel. Alternatively, we'll answer questions at the end of the webinar. Otherwise, we'll reach out to you directly. So for those of you that are unfamiliar with Geneyx, really the company, we have highly skilled individuals with passion for clinical genetics. We average around 15 to 20 years in the clinical workspace, and we have headquarters in both Israel and the United States. Ultimately, our idea is to develop a software that utilizes the genomic and genetic knowledge to ultimately promote kind of a precision medicine and enable a world without undiagnosed diseases. So along those lines, we have a broad implementation. So our software has been adopted by top-tier research institutions, governmental organizations, research settings, clinical and diagnostic labs. We have over 80 customers, over 100 or around 100 publications, and 100,000 of cases that have been analyzed with our software. So our software has been extremely well vetted by the industry, and we really take our customer feedback into consideration when developing our product. Along those same lines, we have a variety of case studies that really show the comprehensive ability of Geneyx analysis. So Bambino Gesù is the largest children's research hospital in Europe, and originally they were analyzing around a dozen cases per month. And through adoption of the Geneyx analysis platform, they were able to scale that up to around 3,000 or 4,000 samples per month with a turnaround time of less than 10 minutes. They also observed an increase in diagnostic yield. As I mentioned earlier, Geneyx analysis is also focusing on rare diseases. So at the Hadassah Medical Center, they were able to identify a unique copy neutral inversion associated with juvenile Parkinson. We also have a role in the somatic workspace. So we've identified novel mutations in colon cancer. And then we also have a case study focusing on how Geneyx analysis is being applied for high throughput diagnostic settings. And so really these case studies offer insight into diverse fields of applications. It also covers personal user experiences. It's easy to read, and we would be happy to send you those available on request. So for those of you that are new to the software, we have a couple different options. We have our Geneyx tests, as well as analysis and then discovery. And ultimately what we're trying to do is automate the diagnostic process from ASQ to VCF and report. The way that we're doing that is we're integrating innovative techniques, machine learning, AI, to develop a platform that analyzes your NGS data but also serves as a resource for discovery. And so what I'm going to focus on today is specifically on the tests and the analysis that we provide, and we'll save discovery for a future webinar. So to talk about the Geneyx test, an excellent way to monetize your lab's genetic data. These are created by our Chief Medical Officer. They're CLIA-CAP approved ready tests and reports that can utilize your existing NGS data or physical samples. So for example, if you want to send in a buccal swab or a blood sample, we have the ability to sequence that and then also provide you with these ready-made reports. And so I just wanted to briefly show these reports because they are highly valuable. So this is an example of a cardiovascular disease screening report. These are entirely customizable, so if you wanted to integrate your company logo or have unique features integrated in the report, you have the capability to do so. The first page is really just a table of contents, but then the next few pages kind of go more in detail in terms of how to use this guide, modes of inheritance. So we cover autosomal dominant diseases, autosomal recessive, X-linked diseases. We have our disclaimer. Now, section 1 is focusing on these cardiovascular conditions, where we cover 374 genes associated with a variety of cardiovascular diseases. Section 2 is very specific to the individual variants that have been observed. So these are a great resource because it'll identify the individual mutations. It'll also provide you an interpretation as well as a disease summary and recommendations. And so in this example, there's actually 3 variants, which we see here. Section 3 is the secondary or incidental findings. So this would be the 73 genes that are recommended by the ACMG to report on. So as you can see here, we have our disease name, our gene symbol, and then the results. So whether it's positive or negative for the individual. Section 4 is the report sign-off. So again, these are CLIA-CAP approved ready tests. This report has been signed off by or can be signed off by our chief medical officer. And then we have our glossary of key terms, as well as the test methodology limitations, as well as general references. So this was just kind of an example report that we provide in terms of the Geneyx test. Again, we do offer a variety of different CLIA-CAP approved ready tests that you can basically utilize off the shelf. So just to really reiterate those value, they're great for established or starting labs. We offer these predefined gene panels and interpretations. They're entirely customizable, and we also have very competitive pricing. So if this is of interest to you, please don't hesitate to reach out, and we'd be happy to provide you with more information. So coming back to the main modules, so we had Geneyx test, but what we're really focusing on is the Geneyx analysis. And so Geneyx analysis is kind of the main interpretation platform. So we have the ability to go in and do kind of a deep diagnosis and biomedical literature search on individual variants, but you can also use this for automation. But what I wanted to do is first just kind of give a general highlight of the main features. So we have a rapid phenotype-driven interpretation. This is really what kind of drives our variant interpretation, especially for variant of unknown significance, or maybe those associated with a rare disease. What this does is it implements machine learning AI-based interpretations and outputs an optimized scoring and prioritization of variants. The way that it does this is it searches across a variety of biomedical knowledge bases and highlights the key search terms that are associated with your individual phenotype. The value of this is we provide you with the literature, so if you wanted to go in and investigate this on a more detailed level, you have the hyperlinks to do so. But again, in terms of kind of an automation workflow, we provide you with the output of the prioritization of those variants, so you can quickly identify those candidate mutations associated with those diseases. We also have the ACMG guidelines. So as many of you are probably familiar with, there are 28 criteria. We automate 18 of those by pulling in relevant annotation sources as well as available variant evidence. This is also a bottleneck for internal bioinformatic workflows. We basically alleviate the complexity of having to stay up to date on these annotation sources. We do that through an automated process, so you can ensure that you're receiving the most relevant information. Alternatively, if you wanted to utilize your user expertise, you can also choose to disagree with specific criteria that are in the ACMG guidelines. And lastly, we are a primary to tertiary analysis. So as opposed to maybe outsourcing your reports, you have the ability to generate your reports within the Geneyx analysis platform. Again, these are entirely customizable. You can incorporate your particular lab design as well as static and dynamic information. So it does serve as a great resource. So this was just kind of a general overview, but what I really wanted to focus today's demonstration on is sample automation. So the first thing that I wanted to focus on was really what are maybe the current challenges with automation. So main issues would be, for example, you have a high sample volume that requires a high throughput environment for analysis. Additionally, maybe you need a platform that reduces manual error, but is also not a black box solution, as well as integration with LIMS, EHR, as well as providing audit trails for management capabilities. So the idea kind of in the NGS workspace is that a lot of this information is siloed and compartmentalized and maybe not fluent. And so what we're trying to do with Geneyx and have done is provide you with a resource that consolidates this information and allows you to actively push and pull data between platforms, but in a consolidated interface. So a primary example is starting with the initial pipeline, which maybe for your company is starting with the FASTQ files. And so what I wanted to do is just show you how this pipeline can be initiated within the Geneyx analysis platform. And then I'll go into a little more details in terms of how that process is being set up. So for the Geneyx analysis platform, for those of you that are maybe new, Geneyx analysis can be installed on the cloud or it can be installed on-premise on your virtual machine. The dashboard view will give you an overview of the analyses that you've created. So these would be VCF samples that have been integrated into a particular protocol that has a filter logic. You can see here we have the ability to go in and analyze the individual cases. You can also search for them as well as assign them. But as mentioned, what I wanted to focus on here is how you can do the FASTQ upload. And really where that's done is through the data management interface. So data management allows you to go in and upload a single sample. Again, this would be more of a manual process, or you can do the batch upload. So if we go into the batch upload interface, what we have here is the ability to apply a TSV file, which I've already created before this webinar. So I'll select that option, and then all you have to do is select process batch. So as you can see, we now have our processing tasks where we have our individual subjects, as well as the pipeline that was initiated, the genome build, as well as the status. And so the status is essentially executing the FASTQ to VCF and BAM file output. And once that process is complete, we'll see that in the VCF sample dialog. So this might take a little bit of time because we are going from FASTQ of an exome to VCF and BAM. So I'll just show you an example output format here. So in this example, the same pipeline was run, and now you can see we have the output of those VCF samples. A nice thing about this capability is we provide really nice audit trails. So if you wanted to go in and investigate a little bit further in terms of relevant details, you can go into the sample here. So we have sequencing sample, which will provide you information and details on the process workflow that was initiated. So if you wanted to ensure that you ran the correct pipeline, from a management capability, you have that option. We also have the processing tasks. So you can see the run that was initiated. It's currently downloading in the status. But once it's ready, it will output the VCF sample. It'll also output quality control data. So for example, if you wanted to look at the mapped, aligned, or paired reads, or for example, look at the coverage profile, we provide information based on 5, 20, and 50x. But ultimately, what we're looking for is the output, which is the VCF file, which again, is just kind of demonstrating the capability within the software. So now that we've kind of shown the initiation of a batch upload using the data management feature within the Geneyx analysis platform, I wanted to briefly give kind of an introduction into how that TSV file was set up. So in terms of the data management, really again to reiterate, what we're doing is we're promoting our users to sequence the FASTQ to VCF and BAM file. We support gene panels, exomes, and whole genome. One of the things that it will require for maybe our current users is the Geneyx data module to be applied to your license. But once this is applied, you have the ability to upload a single or a FASTQ in batch. Additionally, we provide really nice audit trails for that sequencing pipeline. So in terms of the batch upload, really kind of the primary requirement is the TSV file. The TSV file, we actually have a template that is provided. And when you open that up, you are presented with all of the available fields to associate with your particular sample. We have an image on the right, which will show you actually the required fields. And so for each sample, you'd want to enter in that relevant information. Again, we provide you with an example template so you don't have to create this from scratch. Now, one of the other things is there's some specific items within the TSV file that are worth mentioning. One is how you define your target capture regions, and this is actually done through an enrichment kit, as well as the data sources, which is the cloud infrastructure that contains your FASTQ files. So in terms of the enrichment kit, really what this is it's defining your target capture regions for sequencing and the genotype. So we have an enrichment kit that is typically used by default, which sequences or captures all of the exons plus 50 base pairs outside into the intronic regions, as well as ClinVar variants. What that means is if there is a ClinVar variant in an intronic region, it will also sequence and capture that within that enrichment kit. Alternatively, if you wanted to implement your own enrichment kit, you have the ability to perform that customization, but this does require the formats to be in a bed file. If you do perform these customizations, you'll have two options. One is sequencing, and another one is the genotype. So sequencing essentially allows you to define the target capture region, whereas genotype captures specific genetic variants, so that would be individual SNPs. So if we were using this image on the bottom, we have our BAM file plot. We could see that we're capturing specific regions based on the sequencing. And then if we wanted to identify individual variants, we would use a bed file within the genotype. And this would allow you to, for example, output wild type or reference calls. So if you're interested in kind of generating a VCF file, you have the capability to do so. The next item is the data sources. So data sources are essentially your cloud database that contain your FASTQ files. We support a variety of cloud infrastructures, which include FTP-based PACE, S3, OneDrive, and Google Cloud. And ultimately, this allows a secure and rapid communication between the FASTQ files, your cloud database, and the sequence processor. So this is quite intuitive and easy to set up. Again, this is kind of defined in the settings option of Geneyx analysis. But if you have any questions on how to set this up, we have excellent resources, as well as you can reach out to support@geneyx.com. So beyond this, the next idea is the Geneyx analysis API. So a lot of our customers have a LIMS that they want to integrate with the Geneyx analysis platform, and the way that you would essentially do that is using the APIs. And so some of the main functions that this might entail would be to integrate the sequencing machine to automatically send FASTQ files to the Geneyx analysis. Alternatively, if you wanted to integrate the LIMS to add VCF samples, apply protocols, download reports, incorporate patient metadata, you have the option to do so, as well as maybe integrating with your electronic medical records to upload clinical data to patient records. So if a report has been generated and you want to upload that into your LIMS or electronic or EMR, you would have the capability to do so. So as many of you are probably familiar with, Geneyx Analysis is the software provider, so we actually don't have a LIMS on hand. But what I'm going to do is provide you with a demonstration in terms of how that might look like in terms of creating a new case analysis in Geneyx using an API. I'll show you the automatic generation of reports, and then also cover the repository where all of this information is available to our users. Just to preface, some of the main requirements are having a user ID and a user key for the API. Again, this is kind of an additional module that you can add on to your license, and we would provide you with that information as well as the support for getting this set up. So as mentioned, Geneyx Analysis is the software provider, but we don't have a LIMS system. And so what we're going to do is kind of use a modified workflow using Postman. So Postman is a collaboration platform for API development, and in this case, it's going to simulate a LIMS integration platform. So I'll hop back into the software demonstration here. And if I go to open up the Postman, essentially what we have is our user ID, our user key, and then a couple of fields that we're wanting to implement, as well as the protocol to upload these samples into. Now the idea is once a VCF sample has been processed and added into the Geneyx Analysis platform, you can set up triggers within your LIMS systems to automatically initiate the case upload into a particular protocol. So that in terms of automation, this has been demonstrated to show that capability. So all we have to do here is click on the send option, and now we can see that we have a successful code as well as kind of relevant information below here. So if we go into the Geneyx Analysis platform and go to our dashboard setting, we can now see that we have a new sample that has been added to our analysis. In this case, we have our demo RAF2, John Doe, which is just kind of basic. Again, in terms of the audit trail, if we look at this, we can see that we are applying our protocol, which is a cardio panel. We have a variety of genetic models which we've integrated. You can specify the ones, you can customize these, you can incorporate as many genetic models as you want. But then we also have some additional options, including the default genetic model, the disease frequency, as well as the version, as well as if a modification was made to the analysis, you would be presented with that information. Now, you'll notice a couple features here, which include the auto analysis and the analyze. Auto analysis is a great tool, especially for high throughput, maybe diagnostic settings that are focusing on targeted gene panels that just need to output relevant findings that are associated with particular genes. This feature will essentially output a report of all of the filtered variants and give you that capability to essentially investigate it the next day. So the idea would be you initiate the pipeline overnight and then come back the next morning and have that information be displayed to you. So to kind of focus a little bit more on terms of the output, if we go back into our dashboard view, we can open up a process that has already been completed. And so in this example, we have our auto analyze and analyze feature, and we have the downloaded report. Now, before I go into the downloaded report, I just wanted to give a brief introduction into the actual platform in case, let's say you had a genetic expert that wanted to go in and investigate this a little further. That you have the ability to go in and analyze it. And again, this is going to be very targeted because we've implemented a gene panel. So in this case, we can see we have one variant which has been classified as pathogenic by our ACMG guidelines. We also can see we have a very high rank of our phenotype. If we were to, for example, delete our gene panel that's associated with this, we'll be able to see some of those other variants that have that were associated with this sample, as well as kind of the ranking system. So in this example, we have a very high association of this particular gene with the associated phenotype, which actually is the Noonan syndrome. But if we want to look at our phenotyper, we're providing you with all relevant information of the diseases related to this particular gene, which in this case is RAF1. And then we're searching across the different biomedical knowledge bases and highlighting those key search terms. So in this case, we have Noonan syndrome is associated with the RAF1. If we scroll down, we're providing you with the relevant publications, as well as associated gene summaries and publications related to the gene. So kind of a genetic expert perspective, if you wanted to go in and investigate these, we provide you with all relevant hyperlinks out to those publications. So this is the output of the prioritization. So we can see that we captured this one relatively fast in the first line. So this is likely the candidate variant. But in terms of the other features that Geneyx analysis provides, we pull in information in terms of kind of the transcript level, the zygosity, but we also have the asymmetry guidelines. So asymmetry guidelines is very important for germline diseases. And in this case, we can see that it's pathogenic. If we wanted to investigate this a little further, you can see all of the relevant annotation sources that are being populated. So again, the asymmetry guidelines is composed of around 28 asymmetry criteria. We automate 18 of those by pulling in relevant annotation sources and varying evidence. The nice thing about this too is if you hover over a particular criteria, it'll show you what that criteria is looking for, and then also the evidence that was used to apply it. You have the ability to disagree. So if you wanted to incorporate your own expertise and say, I don't want to agree with this, you can unselect that option. You can enter in a particular note, and then this will be stored if you were to save it for all of your colleagues to see in future samples. Additionally, on the right-hand side, this kind of shows you a consolidated view of the information that is being pulled in for the asymmetry guidelines. So we're essentially providing you with all relevant information to make an educated decision, but we're also trying to automate this process, especially for those high throughput environments. Beyond the asymmetry guidelines, we pull in relevant information in terms of the VCF kind of quality of fields. We have evidence being pulled in from ClinVar, so if you wanted to go in and look at those publications again, we're providing you with all of those PubMed IDs to do a very detailed investigation, and it also provides supporting evidence that this particular variant is indeed pathogenic. We have our effect and prediction, and then we also have our frequency, which will show you all of the different databases, including nomad exomes, nomad genomes, for example. So, obviously, if you wanted to eliminate common variants, you have the capability to do so. So the last thing is the reporting. Again, since we chose to do the auto analysis, the report was automatically generated. But if you wanted to go in and create a report for a individual variant, you can use kind of the relevance section. So what this does is it allows you to choose the inheritance model, the relevance, the pathogenicity. You can enter in individual notes. If it requires validation by a colleague, you can choose to enter in, requires validation. But the nice thing about this is it also will pull in relevant information from ClinVar. So you have the options to select the key phenotype as well as the relevant evidence. So here you can see we are displaying all of the relevant information, and what this is doing is it's reducing the manual error that's associated with copy and pasting of that information. So once you are happy with your interpretation, you can save it, and then you can generate that report by clicking on this report icon. Again, these reports are entirely customizable. You have the ability to choose to edit this in the report editor. Again, these can be exported into a JSON, PDF, or even an Excel document, and you can integrate these with your LIMS systems. But if we were to take a look at that PDF document, this is kind of an example of one of our default templates. So in this, we're pulling in relevant patient information, coverage profile, as well as our main findings on this RAF1 variant. We're pulling in a couple fields by default. Again, you can customize this to pull in as much information as you want. But if we scroll down, we now have that clinical significance. So this is the information that we're pulling in from the phenotype and evidence. So here we can see we have disease related to this particular gene, and then we have a nice report that we can provide to the patient as well as integrate within the LIMS system. So coming back to kind of wrap things up, if we go back to our presentation here, so we demonstrated how Geneyx analysis could be integrated within a LIMS system, kind of using an API workflow such as Postman. We demonstrated how we can generate these reports. So the reports are multilingual, fully customizable, and there's also kind of key features that are defined in the protocol. So one of the things that we covered here was the auto analysis. So auto analysis essentially says, for the variants that are remaining from your filter protocol, I want to automatically incorporate those into the report. We have the auto annotate, which includes the user's variant interpretation if available. So let's say a user or one of your colleagues made an interpretation on an individual variant, and you wanted to pull in that interpretation into the report if that variant is observed. This auto annotate will do that, and auto report will automatically generate that report into your account. And so let's say you initiated this pipeline overnight, you could come in the next day and just take a quick look at those reports, validate them, and then provide your electronic signature and sign off on those reports. So in terms of kind of the processing times, so we did just kind of a brief look across FASTQ to VCF files, as well as annotating the VCF files and all the way up to a report. So for panels and exomes, the total amount of time from FASTQ to report is going to be around or under 30 minutes. For genomes, obviously there's a lot more variants to take into consideration. So FASTQ to VCF, this is done under 2 hours, and then annotating is optimized, so it's around under 15 minutes. So in total for, if you're sequencing a whole genome, you can do this in under 3 hours. So kind of together, what this is implying is we have optimized variant annotation for high throughput environments. It's both accurate and scalable, and also promotes kind of a simple integration when you're applying your own internal cloud infrastructures. So in summary, we have a variety of modules that we provide. For this demonstration, we covered the Geneyx tests, which are a great way to monetize your lab's existing data. We covered the Geneyx analysis platform, which, as you saw, has the ability to perform automation as well as go into a detailed investigation and generate those reports. Discovery will be something that we'll focus on later webinars, but it's going to be a great tool for it's going to be a great tool for discovery. So with that said, I would like to open up the panel for questions. Again, if you have any questions, feel free to type them into the chat panel. We can use the remainder of the time to address additional questions. And if we don't get to your questions, I'll make sure to reach out to you directly. So with that said, thank you for attending this webinar, and we hope you found it informative.